Okay, we move on to brain protection. What mechanisms are in place to protect the brain from damage and disease? Because obviously it's a pretty important thing to protect. So here we're going to look at some major mechanisms and then look at some that also will be involved with the spinal cord and then what roles do glial cells play in the process. So let's start with an obvious one. The cranium, of course, the bony skull case is going to be protecting the brain, of course, and remember, there are 8 bones that make up the cranium. So, when you see bone of skull here in this one, that's sort of taking for granted that we're talking about a bony protective layer. And I guess you could throw in skin, scalp as a protective layer. Not very good for protecting the brain, but you'll see some familiar things here. The dura mater, the arachnoid mater, and the pia mater are meninges, and we see the meninges are here, of course, the same as we saw in the spinal cord, and then they're in the same layers. The only difference you see here is the dura mater does have a periosteal and a meningeal layer, and it's just the dura mater actually has a slight difference in its anatomy in the brain. And you see, for example, here the separation, we have this space formed that's actually between the, if you kind of follow it all the way here, the periosteal layer is here and the meningeal layer is here. We have this space called a sinus, and we'll talk about its role in just a moment. But we're going to have the same meninges serving the same protective layers as they did in the spinal cord. Okay, and here we go. Next, the next important protective part of the brain is the cerebral spinal fluid. The cerebral spinal fluid, CSF, is going to be formed in the ventricles. Specifically, it's going to be formed in a place called the choroid plexus of the ventricles. Each of the 4 ventricles, you see one here, one there, the third one, and the fourth one. Each of these ventricles has a choroid plexus. Basically, it's an accumulation of ependymal cells. And you recall ependymal cells are going to be a kind of glial cell. These are going to be specialized to actually use blood plasma to manufacture cerebral spinal fluid. So in the choroid plexus, we're making this fluid, the cerebral spinal fluid, and you make about 500 milliliters a day. And if you look at a typical Coke can, let me put mine over here, this is 355 milliliters. So 12-ounce soft drink can is 355 milliliters. So you're making basically almost 16, 17 ounces of cerebral spinal fluid a day, although you reabsorb most of it. You only have at any one time about 100 or 120 milliliters of cerebral spinal fluid that's going to be in your ventricles or flowing through your spinal cord. So why do we make this fluid? This fluid actually is going to be very helpful in absorbing shock. It actually helps the brain somewhat float inside of the skull. It gives it some buoyancy. It also is going to provide some immunological defense. So this fluid is not just there for decoration. It actually does have several important roles, and we'll talk about where the fluid goes in a second. But again, it's going to be formed in the roofs of the ventricles. It's going to eventually flow, if you kind of follow the arrow here, it's going to flow into the third ventricle and eventually the fourth ventricle, and then it will flow either through the central canal, which actually goes right through the spinal cord, or it'll go around in the subarachnoid space. And remember, the subarachnoid space is going to be between the arachnoid layer, the arachnoid mater, and the pia mater. So we make this fluid, and because we keep pushing it out, keep producing it, we get a pulse pressure that pushes the fluid from the ventricles through the aqueduct, through the fourth ventricle, and down around the spinal cord and through the spinal cord. And we can, so you see here the production of it, and if you kind of follow the arrows, you see the arrows showing it going down through the central canal. You see it going around the spinal cord, and eventually it's going to end up actually exiting the subarachnoid space. And we'll go back a second to this view. It's actually going to end up exiting the subarachnoid space via the arachnoid villus and then eventually re-enter this sinus. the sinus is continuous with our venous blood supply. So, in short, your cerebrospinal fluid will eventually, once it's done circulating around the brain and spinal cord, end up being put back into your blood, being mixed with the blood, and that's how you get rid of it. This just shows you again the ependymal cells, and it also shows you the role that some of these cells have, which is to help serve as a filtering mechanism as well. Sometimes, though, that fluid doesn't leave the way it should, and you end up with a condition called hydroencephalus. And let me type that for you. This term literally means, I can type it, literally means water on the brain. It's not really water on the brain, it's cerebrospinal fluid on the brain, but that's a lot harder to say. So this is how a brain is supposed to look. What you're seeing here, this white thing in a transverse section is showing you the cranium, and the black spots here are actually showing you the ventricles in cross section. Here's when the ventricles are full of cerebrospinal fluid because somehow or for some reason it's not draining. It could be because there's an overproduction of cerebrospinal fluid, or it could be because the small narrow openings that it passes through are plugged up. Perhaps there is trauma, perhaps there is a tumor, and the fluid can't flow through, and it begins to accumulate in the brain. And the problem, of course, is this skull. This protective, nice layer normally is good, but it doesn't expand. So if someone is developing hydroencephalus, the brain can't do anything. It gets squished on the inside of the skull by the increasing amounts of fluid. Now, in some people, this is not really going to be a terribly dangerous issue. It has to be fixed. But for babies, their skulls can expand. Again, they're not sutured in because as they're born, the skull bones are not sutured. So they get this kind of alien look to them, but they actually can have some expansion without any kind of brain damage. Now, for an adult, or really anyone past about the age of 1, when those skull sutures actually knit together, then increasing cerebrospinal fluid pressure can damage the brain. And finally, let me end with the blood-brain barrier here. The blood-brain barrier is a defensive mechanism to keep most things out of the brain. As you see here, normal metabolic changes would cause neuron firing problems. Let's say you ate the wrong thing, or you had something very acidic, or you got a little dehydrated. Any normal changes in the bloodstream, or bag of salty chips, those kind of things would probably interfere with nerve function in the brain, and therefore we can't have that kind of slight change actually be influencing the brain. So the astrocytes, again, these are glial cells that actually wrap around the blood vessels and actually form these very tight junctions, which then do not allow most things to leave the blood and enter the brain. You see up here, if you're too highly charged, if you're an ion, if you're too large or you're not lipid soluble, you can't enter the brain. Some things can pass through. Glucose can pass through. Oxygen can pass through. The things that you would expect that are going to need to be able to pass through the bloodstream into the brain can, but most things cannot. Bacteria typically cannot. Viruses typically cannot. Another problem, a lot of drugs typically cannot. Most of the drugs that we will want to use to treat someone's brain cancer or brain disease can't get through there either. So one of the big challenges in medicine is trying to deliver drugs to the brain, because the brain itself is basically saying, nothing's getting through here. So let me move me out of the way for a second. So the astrocytes and endothelial cells, other capillaries of the brain, help maintain this barrier, and yet we do occasionally still have issues in the brain itself. There are a couple of spots that have to be left exposed for the body to be able to detect and measure changes in the blood, and it's perhaps in these regions that we are getting some viral or bacterial invasion of the brain, and so you can get things like meningitis because of the occasional infection of certain pathogens may be able to escape the blood-brain barrier. But again, I want to stress this is not like a membrane or a sheet. It's the astrocytes, which are protecting to make sure that most things cannot pass into the brain so that it's not going to be interfered with because of its critical function. That does our video on protection for the brain.